Diversity, I think it's essential.
In the context of a farm, you can think of planned biodiversity and associated biodiversity. Associated biodiversity typically reflects the planned biodiversity that the farmer made. So if a farmer decides to keep larger extents of natural landscapes, whether it's forest or grasslands, you'll typically see birds or other wildlife or insects or plants associated with more of those natural or perceived native habitats move in and use the farm. So associated biodiversity can include wildlife like birds, deer, squirrels, insects, can include plants. You can also think of it as some of the associated ecosystems that maybe aren't managed as part of the farm, so riparian areas or field buffers. Typically they're a little more wild. You can think of it as novel ecosystems or managed ecosystems depending on what community composition they represent. Typically, but not always, they don't mirror the native vegetation or native communities exactly. You might have some plants that are invasive or non-native, some birds from other ecosystems that might not be expected. For example, at Greenbrier farms that where we see the cattle, we have a lot of grassland birds. That really reflects the change of the landscape to farmland from forested.
Some of the things that I've noticed as the garden has developed over time is that we're now seeing a lot of new species in the garden. And that's not just plants, that's insects, especially dragonflies, bumblebees, honeybees. Seeing a lot of pollinators come into the garden. This year with Katie's work, we're starting to see a number of birds come into the garden. And that's really important for this space. If you can envision that originally this whole triangle here was grass. That's all that was out here. And now we have a lot of diversity brought into this area. And so we have the insects coming in. We have the birds coming in. And so this is a much more powerful space now that that's occurring.
I've really noticed a much higher diversity and abundance of bird species in gardens that are very diverse both with type of plant and the structure of the garden. Since I started my project here with the garden, we've noticed that we've seen more birds of the same species and also new species starting to come into the garden, which is really, really exciting. So first, before I started, we really had just a few goldfinches and some bluebirds in the bluebird boxes. But since we've started, we've seen an increase in both bluebirds and goldfinches, and we've seen robins enjoying the blueberries that we planted, as well as some song sparrows and northern mockingbirds. Because you can see where the garden is at. It's in a large grassy area in between two spots with trees. So for birds to get from one spot to the other or into the garden and out, they feel sort of unprotected when it's just a large grassy area. So adding the larger 3D structures help provide some protection for them so they can come enjoy the garden and not feel unsafe.
Probably the most important thing that I've found, a new species that I'm seeing in the garden, are Homo sapiens. I now have students who will come and walk by and talk to me about what's going on in this garden. Just a while ago, I had a freshman student walk up and say, "I would like to work in your garden." I think that's the most exciting thing, is we now have something visual for students to see, and it's bringing interest into the garden. And more students are appearing who want to work in botany and want to work in the garden setting.
We got weeds down here. I mean, it's phenomenal. Right now, they're terrible in my orchard. Terrible. I think crabgrass grows 6 inches a day. And morning glory, faster. Yeah, weeds are a problem. So it bothers me. It has bothered me how much chemical I have to put on. We got a lot of water, lots of sunshine. Why can't we rejuvenate agriculture in the South?
During the winter, we have the ground team, and they vacuum up just tons and tons and tons of leaves. I will direct those guys to channel the leaves to a certain part of campus and just let them compost and cook all winter and spring and summer. And then the next year I go to those piles, which are like about 9 feet high, huge, and they decompose. It's turned into what we call leaf mold. And at that point, it is loaded with microbial activity. 1 teaspoonful of this would contain upwards of 8 billion microbes. Yes, microbes, they're put here on Earth. They're short-lived in a way, but they're here to do one thing: eat, poop, and reproduce all day long. That's all they do. So the more ideal that you make their environment, the faster that they will do that: eat, poop, and reproduce. When you see wood chips or you see a compost pile or whatever and it's steaming, that is actually what? What is it actually? Activity. It shows that it's active. It's got lots and lots of activity there, and it's really showing that it's ideal because they're doing what they do, and they're doing it at rapid speed. And it's doing it so fast until that activity is producing heat. So it really has nothing to do with the ambient temperature. It's got everything to do with what's inside the pot. Okay?
So my name is Min-Ken Liao. I'm a professor of biology at Furman University. I'm a microbiologist. My student researcher and I are interested in bacteria in water. In the past 15 years, we've been studying bacteria in water. And just in the past 2 summers, we started to study bacteria in soil. Now, the reason we're interested in soil is a handful of soil has more individual microbes than all people have ever lived on Earth. So that is an abundance. But what we're interested in is diversity. So our overarching hypothesis is that different soil environment will support different microbial communities. So different soil environment, including different land management, like we have a forested area behind a chapel, nobody's managing that environment. And we also have very beautiful, well-maintained lawn that is chemically managed. And we also have Furman Farm, which is organically managed. Now, different land management could also mean the different root systems created by different crops at Furman Farm. So what we started with last summer is to study different soil samples on campus. And we took 2 approaches. One is culture method. The culture method is we grow bacteria on the plate in the lab environment, laboratory environment, and we see the differences between bacteria that grow from different soil samples. Now, to see is to believe, so that is a good approach. If we see differences, then we say there are different microbial communities. And what is the advantage of the cultural method is less than 10% of the microbe can be grown in the lab. So we're missing out a lot. So we have to use another approach. The other approach is non-cultural method. Basically, we just study the DNA instead of growing the bacteria. What we do is we collect some soil sample and we add some phosphorus solution in the soil sample. We shake it really, really hard to release the bacteria that are attached to the soil particles. Now, once the bacteria are out, we collect those bacteria and we burst them open, so all the DNA are coming out. Now, lucky for us, different species of bacteria has their own signature genes. So we just harvest different signature genes because those signature genes represent different species. And we analyze those signature genes to find out who they are. Now, our results, based on the culture method, is that we can actually see different microbes, different bacteria. We saw more bacteria, different kinds of bacteria from Furman Farm soil. So by culture method, Furman Farm has great diversity. Now for the non-cultural method though, the jury is still out. We don't know, we cannot even conclude, we cannot even confidently say that Furman Farm does have more diverse bacterial community. It seems like the very poor environment has just as much diversity.
Each row that we actually go to in the garden, we like to have worm activity down each and every row, even to the point per square inch. We like to see about 10 or 12 minimum worms per square foot, per square foot. Because if you don't, by textbook standards, you need to work your soil until you can see that. So it's not really what's going on above the ground, but it's all about what's going on beneath it. And if you have those worms in place, your bacteria is sure to be there. It's a promise.
So the most important part of my research that I've really taken away from this is, there's a huge body of literature about urban ecosystems and how we develop the land affects our environment. And we all have houses or just a small plot of land somewhere. So what I've really taken away from this is, what can I do to help increase biodiversity on my small little plot of land? And I've really taken away that you can help diversity just by, we just planted a few blueberry plants and put some structures in, and all of a sudden we have 3 new species of birds. And so that's really, really encouraging to know that even within a very urban area like we are here at Furman, that you can still make a difference.